So today, actually, this topic is a little bit outside of the main, homework and kind of task that you have. But the idea is really to touch upon something that we don't cover much, not in the exercise, but I think it takes much of us as biologists in the real life, which is understanding not only the, static aspect of molecular biology, which is genes and domains and proteins and so on and so forth, but really address the expression aspect of it. So in order to do this and not really disconnect myself from the theme of the class, I'll mainly talk about genetic effect on gene expression across human genome, and I'll illustrate maybe two and a half sites that I think are really critical, and it's important that any biologist or any whatever data scientist of biology or any of you will be familiar with. The first one is GTEx, which is the name is for genotype tissue expression. The second one, which is Human Proteome Atlas. And the third one is mainly just to remind you that there are ongoing effort in Europe, mainly to, I would say, to keep pace with what used to be only in the NIH type of resources for gene expression that I'm sure that most of you are familiar with, which is GEO. So everybody is using it. I want to mention that there is a complementary, very good one that is building up. It's not as exhaustive as the GEO, but I think in many aspects is giving much more interesting information. So this is just as a reminder. So let's start with both of them. So the motivation is rather clear, and I want to mention it in view of the genomics, not to forget about the connection between genomics and gene expression. So understanding, just the complex trait genetics means to, first of all, to know what's written in the genome. And for that, you will deal quite heavily with SNP and variants and all those aspects of haplotypes and VCF and so on. So this is what's written here. And the second, part, which is as important, is how the fact that A is here will affect what the cell is doing, what the tissue is doing, how the pathway is going on, and so on. So those are, of course, heavily connected. I don't have to tell you this. Now, the majority, this is also something that often is not said loud and clear. The majority of trait-associated variations in our genome has nothing to do with the coding. It's mostly in the regulation, amount-wise. No, I'm not saying who is more important or less important. I'm just saying by amounts. So the common hypothesis is that most of this function that we are talking here will be altered by gene expression. That was the reason to combine the two, I would say, major paths of the genomic revolution. One is understand what's written in the genome, and the second is what's written in our transcriptome. Now, in the old days, and when I say old days is maybe 15 years, most of what we knew about the transcriptome came from pre-decided decision on RNA chip and so on, Affymetrix chip and so on. So a list of what we expect, and then you can say it's up and down and so on and so forth. The revolution is that by now, most of what we get from real transcriptomes comes from RNA-seq, which is this is just a reminder for me to say that it's really high accuracy those days, extremely high sensitive. So for the first time, you can see event, let's say gene expression event, that you couldn't see before because of sensitivity. And when I say sensitivity, of course, it's also exposed what we didn't know much before, our non-coding RNA, long-coding, non-coding RNA, alternative splicing, and so on. And all this introduction is just to mention that GTEx took the effort in combining those. So what's the idea? In a very schematic way, we are looking for EQTL, which is by, the name is expression quantitative trait locus, but the more, let's say, intuitive naming is to try to find association between genotyping, which is those letters that are written in our genome, and RNA expression level. And I think it's all illustrated in this very simplified version, in which you have here a gene. Let's say this is the transcription start site. This is a SNP that could be fairly far away, doesn't have to be in the promoter, doesn't have even to be in the immediate promoter or remote promoter. It could be rather far away. However, whenever you look at population or individual with A, you get this level of reads. Whenever you have C in this same position, you have this level. Or in other way, let's say in the box plot that you just saw, CC will give this level of expression, CA will give this, and so on. Okay, so that's the idea. That's what we are looking for. Please.
Is it like a mere correlation, or is it something that we know of or suspect of someone straight effect?
It's all, so it's, first of all, it's all by correlation and statistics. And I should say that the level of false positives and false negatives have been estimated in their papers of GTEx, which is initially was quite high. And recently, the new version that came up include, I'll go through some of those numbers, but it's a good question, of course. And also, there are some definitions that I'll go through, so you'll see that it's rather sensitive to what you ask, of course. So the project started in 2013, so pretty late. And by now, we have version 8. As you can see, it's really very fast evolving. And the idea was to characterize human transcriptome within and across a wide variety of primary tissue and cell types. Now, the first pilot, which is what you have seen till a month ago, included 450 human donors, post-mortem, of course, in which 44 tissue were extracted, crushed, and RNA-seq was performed for each and every tissue, healthy tissue. Those are healthy donors. And the pilot include 1,600 of those RNA-seq data set. Full transcriptome, one tissue, many, for each tissue, many donors. And you have this reference that is. So again, what is the GTEx show us? It's not a great resolution, but we have donor 1, donor 2, donor 3. Let's say we have 3 tissues, the liver, the heart, and the brain. And what we have here is AA, CA, or CC, and you have this changes. So for example, in the liver, no change, okay? So same amount, either you have CA or it doesn't care what you have in your genome. That's an important comment. In the other tissue, again, never mind what's the tissue, what's the level. Could be high or low, but it's not differential between the tissues. But look what happened in this, in the brain. You have a very high sensitivity to what's written there, right? If it's a heterozygous, or if it's a homozygous of this type or of this type, you have a very big difference in expression. And this is exactly what we're trying to capture. So in the histogram will show you what you expect, and this is what the system showed. So just to summarize, what are those samples? First of all, it's a known disease, and that's important because in the other resource that I'm showing you, there will be also pathological tissues, and the difference are very important. So this is non-disease tissue, normal range of age, whatever it is. I mean, I don't know what it is exactly. But anyway, it's the 44 sample. And the minimal to be represented by now in GTEx is that at least 70 donors will contribute to the analysis. So this is a little bit like answering some of your question. So 31 solid organ, 10 brain sub-region. Actually, it's 8. 2 they took and did duplication just to check themselves internally how well. So this is part of their, so what we see is also the quality control of them, but that's fine. Actually, I like the fact that they represent it as part of the data and not hiding that somehow. So this is 8 sub-region. Yeah, duplication of the whole blood, which is quite terrible definition. What's whole blood? I mean, again, but that's because it's quite informative. We'll go in the. I don't think it's a good choice, but anyway, and 2 cell lines derived from blood and skin samples. So they wanted to do this, and again, 80 like we see in many genomics studies. Many of them, most of them, 85% are European. So we do not have a full representation of the genomics related to transcriptomic in other ethnic group. Okay. So the aim of the study is really to get 2 effects. One is what I described as cis EQTL, and when I say cis means in the same chromosome, in the same DNA strain, okay? And trans, they hope to, maybe I'm a bit too critical about GTEx. It's a great resource. So I'm just saying that the trans, in my view, is useless. Despite the fact that they present it, I looked at it, it's very hard with the amount of data that they have to really find good correlation for trans. It's just statistically very hard. It's not that they did something wrong. It just has to wait for much more data in order to convince that something is trans. So we won't talk about it. We won't even discuss it, okay? So, but the cis element are really very powerful. And the example that I'll show you comes from GTEx 6, which was about a year ago, and a big Nature paper that described it all. So this is the 44 kind of tissues. And what they have is the total gene that showed the scheme that I showed you in the graph, namely that they do have an expression-dependent tissue that will differentiate between other tissue. So you can say that it's a cis-trans tissue specificity somehow. They have with FDR of 5%, 20,000 event. The trans is, as I said, is nothing. It's 100, but I think it's 10%. I mean, it's not that strong. If you take one tissue and say what is the maximal number of EQTL in this specific tissue, you have this nerve that has so many donors. This is the number of donors, and they have 8,000 EQTL, which is a huge number. And the trans, forget it. Okay. So just to summarize, so you understand how they build it, because once you understand the build-up of this, we can understand what we are going to see. So this is the summary. Yeah, it's all there. And this is the 449 individual. This is the tissue. I'm showing you it because it's a Python visualization. So it's a continuation to the class that we just learned. So you can see that it's pretty nice visual because all the 44 tissues are here. And immediately, without saying much, you immediately can see that some tissues, and this is just the genome. This is the RNA-seq. You can see that some genome were very heavily covered, some are really poorly. And in order to do it in a different visualization, I just show you these 44 type of tissues. And here you see how many contributed to this tissue. So for example, adipose 300 contributed to the tissue. This is some quality control, so forget about it. And you can see that, for example, in each part of the brain, they didn't have so many it's about 100 or less. So immediately you see that they had to deal with statistical, internal, non-identical statistical power for each. But this is well done. It's very professionally done. So this is it, and this is just to show that the numbers are very good. One thing is that they define, I'm not crazy about this definition, but I think it's the accepted one, that to have a EQTL or to have a cis candidate, you should be within 1 million from the TSS, from the beginning of the gene, not from the end. So it's some kind of accepted value for what could be, let's say, still in a regulatory unit. And that's their definition. Okay. So what else? And this is just the end of it, saying that once they did all what I just told you, RNA-seq and so on, they can start looking at events that were completely overlooked before. For example, cases in which allele-specific, you can see expression only from one allele. Why they can do it? Because you can look at the imbalance of RNA-seq reads between allele in any heterologous site. If you have C or A in the same site, you can see that the reads that comes with the C are 100 times, and the one with the A is 0. So for the first time, you can say, wow, this is a allele that is only expressed from one of the strand or of the, of the chromosome and not the other, which is quite unique. Another thing that they did only recently is because of these powerful tools, they can say something about alternative splicing, which is tissue specificity. You see why? Because for the same reason, you can look and say, in this tissue, do I have this junction between exon 1 to exon 5? Yes or no? If yes, how much? And how much is it different from any other tissue? So they called another unit that instead of E gene, which is expressed gene, S gene, which is splicing gene. Okay, so this is the two terms that are now used. So that's it. Just to say, to remind from the biology, because I'm going to do the illustration, is that despite the fact that we are often like to talk about the molecular level, we should remember that a lot of events occurring at the cellular level, at the tissue level, at the organ level, and the organism level. And we cannot forget it when we do this molecular game. Okay, so let's go to the illustration. This is just for you to have another source of gene expression, very good resource, the one that you all know, GEO, the dbGaP, that is quite a pain to get it from the NIH, but once you get it, it's very useful. This is usually genotype and personalized identifiers, and this is the body map of Illumina, which is also a nice source that I don't know how many are familiar with. So what the project is expanded from what I showed you is that the sample size really increased. By now, it reached 1,000 donors and 53 tissues, again, something like that. Computation and experimental method became really nice. Visualization became tremendously improved. And they are planning, they don't have it yet, analysis of single tissue, or you can combine because of your own idea that you believe that this tissue and this tissue and this tissue should be analyzed combined. So they will give you the opportunity to do it. And inclusion of genomic annotation is not yet there. So let me go for the illustration. Okay. Are we working with this or with this? Okay, that's good. Okay, so this is their website. No. In most places, it just jumped to you directly. Okay. GTEx. Where is the... Where is the... This is the article. No one is using it, no? I don't... Okay, where is the link? Nadav, can you come and just find me the GTEx, the real GTEx? So, yeah, just to save some time. So what I want to show you is the new, yeah, somehow. I don't think it's from here. It's not the one that usually I'm getting in.
We're using Bing, which is...
Yeah. They don't. That's the problem. Oh, okay. Now it makes sense. Okay, Been never heard about most of what's going on in this world. Thank you. Yeah. Thanks to Google. Yeah. Okay. Yeah. That's the web. Yeah, we can make it. Yeah, exactly. So to explore GTEx, they have like in many other cases, you can go through your gene of your favorite gene. You can do by variant, because remember, we are looking for the genomics aspect of it. It's not just the gene. You can do it by the tissue. You can do it through histology images, and they provide you the histology. And they do also have already some kind of a meta-analysis. For example, you can do a query of multi-gene, because you just get a list of genes, and you want to get it together. You get the top 50 genes for each tissue. You can have a nice transcript browser that we'll go over. And they do have everything that looks like a browser that is only dedicated to eQTL. So despite the fact that we told you many times, use UCSC whenever you want, I would definitely prefer to use this for the expression, because this is a cleaner, nicer, and more focused for expression. And then they have, as I said, a lot of things that are coming soon that they are not there. And just as a biologist, if you find something from a specific donor that you want to invest, you can come back to them, because they have a biobank that they will be willing to send you the sample if needed by really rather minimal funding is really good. So they really, I want to mention it because it's an open source, extremely good one. So and runs by professionals. So let's go through. Let's start with top express gene, just as an example. So you can go and immediately see the 44 cases that they have. Let's go to adipocytes, and immediately you see this version of, again, I'm not crazy about it, but immediately you can see that this version gives you the list of genes, and they are already clustered. Those are the 50 highest expressed genes. So if you just want, for example, as a biologist, not even to know what is the difference, what EQTL, but what will be a good marker for my tissue, just go there. And immediately you have your selected choice. So it's very useful for other, what I'm trying to tell you, that this source, in addition to the very fundamental aspect of EQTL that we just, is a very good source for many other things that we need as a biologist. So this is just, let's go back and let's go to a gene. Let's go to something creative. Do you have a nice gene that you want to look at? Let's see. Yes. Let's see. STX BP1. I have no idea what will come out, but this is a gene that I kind of okay. So this is a gene that is syntaxin binding protein. It's very important in the brain. So immediately you can see that indeed it's the yellow is the brain. It's indeed very prominent in the brain, and you can see the expression in a violin plot, as Nadav showed you, with a very informative information. For example, how many donors help to get this plot, the average, the mean, and so on. Quite a lot of information. They did something nice in which you can look at, for example, if you have a difference in sex, which is not expected always, but you learn a lot by looking at, for example, male and female now separately, and you can see that the expression is 300 here, but in male is 380, which is quite important differences, and so on. So this is one aspect which is kind of nice. You can do it with a log scale or not, and immediately you see that what looked to be very little, it's actually expressed in a reasonable amount, but just in a log scale, you can see that, of course, the brain is dominating. You can, of course, do all kind of just nice visualization by sorting it by tissue sort or by median or remove the outliers and so on. Okay, so you can have a very nice, and because they wanted it to be publishable, they allow you a tool here to do the filtering here on the figure. So if you just want the brain or just want, so just you do it. So it's very easy to do the filters and decide what you want, what you don't want, okay, from the picture. So this is just an overall. Now, once we do, I closed it, yeah. I didn't mean to, but okay, let's go back. Sorry. Okay, once you do, yes, please. Right. But by now, I just looked at a gene, specific gene, and asked how this gene is expressed in every donor that provided the information throughout 44 tissues. That's what I showed you as of now. But I didn't show you anything about the genomic and about how good it is and so on.
But I'm asking if they're planning to go to the resolution of cell types, not tissues.
It's not, they are planning to. I wouldn't use this site. I'll show you a better sites for this.
I think they do have some cell lines.
Sorry?
I think they do have some cell lines, like not many of them.
Yeah, they do have some cell lines. I'll show you the connection with another illustration. But let's look at something which is kind of nice, the browser, as I mentioned before. So if you look at the browser, at their browser, you immediately see the gene. Again, you can see, let's see, not select all tissue, just let's say take the adipocytes. You can choose any of your choice, and immediately you see the browser in a fairly non-classical way. Let's see the following. What you see is only the EQTL of, in this case, 2 types of adipocytes. And whenever you have and this is the area of the gene of interest, right? This is the gene that we talked about, this, right? So what you can see is, first of all, that those are EQTL. Each and every one is clickable. So you can see which variant, which encode size, what is the gene, what's the p-value, how strong is the EQTL in this specific tissue, what is the SNP, and so on and so forth, for each and every spot here. And you can, of course, zoom in, zoom out as much as you want. By now, I'm looking at 200,000, like 250,000, okay, nucleotide. So what you see here is, again, they provide you the RNA-seq data. They provide you the EQTL analysis for variant, and you can start and see how those fits. I mean, really, the raw data. What they added here is also what the word is telling us about the association to disease and traits. So this is the GWAS catalog. Did they heard about GWAS catalog as of now? We didn't mention it. Yeah. So GWAS catalog. You are all familiar with GWAS catalog? So just I'll say it in one word because I just wanted to show you that they do have integration with information on genomics, global information. GWAS catalog is a catalog of any experiment that was done with cases and control for any trait that you can think of, such as, let's say, obesity, BMI, depression, or whatever. What is, if any, variant in our genome is associated statistically to this trait? Okay? So you get the feeling? I mean, I'm saying it in a very naive way, but basically is provide the word current information on association of variants with traits. So they show us all their information in view of what is known as of now on variants and traits. So if I'm seeing this, I can stand on this and say, okay, what is this? And they tell me that this is education attainment. So this is a phenotype of how many years you were in school. This specific variant tells you if you had to live at 10th grade or 12th grade or 6th grade or whatever, but might be of interest if this phenotype is somewhat associated with the brain function and so on. So you start to build hypotheses by looking at such an integrative way of looking at it. I love it. I must say that it's often you can learn a lot out of this search. And so this is just to show you this type of information. And so this is I just looked now on the adipocytes of this specific one. I want to go back maybe to the other part of the game here. The expression I already show you. So the expression, the search gene, we already went through. The transcript browser, I didn't show you, but you can look at the top one I showed you, the multi. But I want to show you now another way of looking at it, which is not the calculator. Let's go back to the portal, and let's go back to the, yeah. Let's go back to the gene of interest, because that's a good way to look for. So STX-BP1, I want to show you that once you get this gene, you can go immediately to you can go to this level, okay, that I showed you. Now you can go to the level of what's the exon expression. Now the exon expression is shown by which in each of the 44 tissue, where is the maximal expression according to exon 1, 2, 3, 4. So the size of the exon gone, it's only the numbering of the exons. So again, I think visually you immediately see that, for example, this exon is very highly expressed versus this one. Anyone have some idea why this exon is much more expressed than the others? Yeah, exactly. Is of course artificial of the methodology, because they did poly A and so on, so they have a gradient. So don't take the value as, you know, as important. It's not the value, but what you can learn is what is the difference between the different tissues in view of this aspect and on alternative splicing. So this is one aspect. This is really the graph of all the exons that they see in these genes. And then you can go and say, what is the significant single tissue for this, whether you have a single tissue for these genes. So immediately you see that, yes, they have a p-value that is not that great, but yeah, the p-value is not that great. So apparently there is nothing that in this tissue has a unique EQTL that is a single tissue event. So you can, of course, it doesn't say that it's not a multiple tissue event, so you have all the statistics here, and then you can say, you can go actually and see significant, that we already have been. I want to show you something else here. You can look at the junction expression, which tells you in the exons, now they just focus on the RNA that is on connecting two exons, which is beautiful. So now they only look at alternative splicing event. From that, they can learn which gene is specifically spliced in what tissue in a surprising way. Okay? Those are the S gene. So this is nice way. This is a new addition. And then the isoforms themselves, which is, again, how many isoforms you have, and so on and so forth. Again, it's a version of the same idea. So do you have another energy for another 10 minutes of beautiful, beautiful site? OK, great. So we have a new student. OK, so from now, I want to go. So I hope we are still in the Google search. So human proteome atlas. OK. The Human Proteome Atlas is a beautiful atlas that is developed at 2003 by the Sweden group. They really, I'm following this project from 2003. So I can tell you that it's a very creative idea in which they say for the first time, let's look what happened at the protein rather than the RNA per se. And in order to do this, they had to use antibody, specific antibody, for each and every one of the 20,000 gene. Between 2003 to 2007, they bought every antibody on Earth and realized that 85% of them should go to the garbage. So this is an important message, really, that most of what the antibody that are commercial available would not have the quality that is needed, especially for doing something really global, maybe when you look for your protein. So what they had to do is to develop a new method to develop new antibodies that are now can be bought by Sigma. It's called pressed antibody, that are shown with a very high quality. And after finishing this, and after having about 250 pathologists from India actually working with them to understand what they see in their tissues and so on, they built a curated version of every tissue and organ and cell line that you can think of. And this is the new version. And it's ongoing project, very nice one. So let me just take you very briefly into some of those. I'm taking it from the introduction just to make it very efficient. But one aspect of it is the tissue atlas. As I said, so to look at the tissue atlas, wait, okay, we'll go there, okay. So immediately you have a summary on the tissue. You get it's like a green light, which says it's good, it's high quality. If it's not high quality, they mark it as a poor quality. So immediately you see that this is the antibody that works, and you have everything that is on this side will be RNA expression, and here a score of protein expression. And in this specific case, you can see that only muscle tissue are expressed. So this is a unique case in which this is what you see here is expression value only in muscle. They compare it already with RNA expression, which is also only muscle. They put also in their own database, they include the GTEx, as I showed you, result. So you can compare other resources, such as the GTEx, and they include the Phantom 5, which is an excellent Japanese resource. So you have immediately everybody in the world that looked at it gives, in this case, consistent result. It's only muscle in a specific muscle, okay? So this is the tissue. Of course, you can go and immediately learn about which antibody were used. When you look at the antibody, you see the result of each and every antibody that were used. What is the quality? What is the dilution to use? What is the Western look like? And so on. So you have a very, and their pressed analysis. You have a very good, and they give you really what cross-reactivity you might have with this specific antibody. Very important resource on this. So again, you can go to the quality, but I want to show you now the cell level of this. Maybe it's not that great. Okay, let's go to another example. This is the cell level. I'll show you this and I'll stop here. The cell level is showing where protein is expressed with this antibody that we show all the quality control in a cell. And in order to do this, what they do is they have human cell, they have RNA expression, they have, so let's look at their beautiful, microscopy. You can only look what happened if you stain the nucleus. You can add the microtubules if you want. And then you can add the antibody, or you can add your antibody and the nucleus. And you can, of course, also see the border of the cells and so on. And immediately you get what is the expression level calculated from this in several repetition, in several cell line. You can see the cell line and the antibody. So different antibody, whether they give different result, the answer is yes. You can see this. And in addition, they do also specialized experiment. For example, how will it be in cells that are divided or not divided and so on. So it's a very rich resource for cell view. And again, I think immediately you can see that this specific genes is expressed in a time point. So this is cycling only when the cell is about to divide. So you immediately can calculate stuff on and so on and so forth. So just to show you that, I'll stop here and just summarize. They do have also only recently brain resolution, very nicely done. Only 5 regions, not highly resoluted, but good for, I would say, kind of not very detailed brain analysis. They do have a new stuff, which is cell line. So 69 of the major cell lines, they have this information, detailed information. And what I find the most attractive in this site is that it's extremely well connected to the major resources. So if you don't like what they show, you can immediately go and see how the others are expressing to the same level, and it's any picture here is manually curated by experts. So nothing here is just done automatically.